Hello, good morning, teacher and friend. In this video, I will show you or I will present to you about classification of viral genome sequence using machine learning methods. My name is Israel. My NIM number is 2602 1061. Introduction. Rapid and accurate identification of virus is crucial for effectively managing infection. This is viral genome sequence offer valuable insight into viral characteristics, aiding in tracking their spread and developing efficient control strategies. However, traditional methods of analyzing the sequence are often complex and time-consuming. This is where machine learning steps in, offering a powerful tool for viral genome sequence classification. Among various machine learning architecture, convolutional neural networks (CNNs) have emerged as a preferred choice for this task. State of art. Several studies state of Several studies have showcased the effectiveness of CNNs in classifying virus, including SARS-CoV-2 or COVID-19 and influenza, based on their genomic sequence. Pacific is end-to-end tool that employs a CNN with a architecture and data argumentation to classify 5 DNA virus, SARS-CoV-2, other coronavirus, metapneumovirus, rhinovirus, and influenza. Research until this a simple CNN architecture with data argumentation technique. Data augmentation artificially generating various the existing data. Introduction, random mutation, and insertion of deletion to increase diversity of training set, improve the model's ability to analyze and data. One study proposed a CNN-based approach named virus to fact for classifying virus based on their spike protein sequence. The spike protein is a crucial structure on the surface of many viruses that plays a crucial role in facilitating viral entry into host cells.
Material and method. For this study, we obtained datasets from the National Center for Biotechnology Information virus database of viral sequences. The table on the right shows the distribution of samples used in the study. We obtained 4,000 samples of SARS-CoV-2 genome sequences, 4,000 samples of influenza virus genome sequences, and 4,000 samples of random viral genome sequences, for a total of 12,000 samples. Each of the different classes were given a label, one for SARS-CoV-2 samples, two for influenza virus samples, and three for random or control samples. The sequences are then encoded into a form that can be fed into the proposed machine learning algorithm. For this study, we use one-hot encoding to represent each letter of the sequence into a string of one-hot vectors. The machine learning algorithm that our study uses is a convolutional neural network, or CNN. CNN have been used in a variety of machine learning-based applications as they are fast and efficient in terms of representation of text and sequences. CNNs are composed of an input layer that accepts a tensor, an output layer, and one or more hidden layers, such as a convolution layer, fully corrected layers, or normalization layers. We use five metrics to evaluate the performance of our machine learning algorithm. Accuracy measures the ratio of true instances to the number of total instances. Precision measures the ratio of true positive to the number of true instances. Recall measures the ratio of true positive to the number of true positive and false negatives. Specificity measures the ratio of true negatives that the model correctly predicted. However, in machine learning, there exists a trade-off between precision and recall, where an increase in one metric would reduce the other. We use F1 score, which measures the harmonic mean between precision and recall, to balance these metrics. To further gauge the proposed model's performance better, we also built four more separate machine learning algorithms to compare its performance to. We use support vector machine, CNN BILSTM, k nearest neighbors, and random forests. This graph shows the implementation of our proposed machine learning algorithm. First, we collect samples from NCBI. Then we combine the samples together and encode them into one-hot vectors. The samples are then shuffled before being fed into the machine learning algorithm. We use a tenfold cross-validation scheme to train the algorithm. Each fold, 90% of the sequences are used for training, while the remaining 10% are used for validation. We then record the performance of the machine learning algorithm using the aforementioned 5 metrics. This graph shows the implementation of the CNN-based machine learning algorithm that we use. The network consists of one input layer, one max pooling layer, one convolutional 1D layer, flattened layer, one dense layer, and one output layer, which consists of three neurons, one for each class of sequences. Result. The proposed CNN-based machine learning algorithm obtained a tenfold average accuracy of 0.82891 with a specificity of 0.86632 and F1 score of 0.82121. The machine learning algorithm scored a higher precision score than a recall score at an average precision of 0.84146 and recall of 0.80255. The machine learning algorithm's performance remains stable at the end of each fall. Among the four other models that we tested, support vector machines scored the highest average precision, 0.93938, while scoring the lowest recall at 0.47245. On the other hand, CNN BI-LSTM scored the highest average recall at 0.89161, while scoring the lowest precision at 0.56786. Random forest scored the highest accuracy and F1 score at 0.87100 and 0.87130, respectively, indicating that the random forest model is very effective at viral genome classification and strikes a good balance between precision and recall. Conclusion. Quick and reliable identification of viral sequences is critical as infectious disease continue to pose significant global challenges. Machine learning-based approaches offer a powerful tool for viral genome sequencing. This study demonstrates the use of convolutional neural networks in classifying SARS-CoV-2 and influenza viruses based on their genome sequences. We achieve a great result of an average accuracy of 0.82891 with a specificity of 0.86632 and an F1 score of 0.82121. Other machine learning based models were also tested, which yielded comparable results, demonstrating the capabilities of machine learning algorithms to offer rapid and accurate identification of viruses. Thank you for listening to our presentation of our research. We apologize for any mistakes or inaccuracies. Thank you.